Hey guys, I was looking through some sediment samples that I collected from a nearby estuary flowing out to sea, and I came across something pretty unexpected. This is something that I've seen in the literature, but I've never seen up close before, and if I'm not mistaken, I believe this is a veil, a large structure made by this particular bacterium called Thiovolum majus. It is one of the fastest moving bacteria ever recorded. That is when it's not tethered to a surface by a stalk. I can't remember exactly how many body links per second. Feel free to correct me in the comments, but I believe it's something like 65 body links per second. And what they do essentially is they come together, they aggregate by using these specialized stalks, which are essentially made from all these protein filaments that are interwoven together. And they all aggregate and they attach themselves to a surface right at the border between where oxygen from surface waters diffuses downward and sulfide diffuses upward. And by creating a current, by spinning their flagella all together in unison, they're able to drag the sulfide from deeper sediments towards them upwards and the oxygen downwards towards them. It's essentially a tractor beam. And there are other organisms that appear to be exploiting this too. And hopefully we'll get like a nice view of those in a moment. So this is a really cool example of prokaryotes coming together and creating a structure that creates essentially new habitats for other microbes. It's kind of like the microbial equivalent of a beaver dam. So it's a little bit hard to see. I apologize for the footage being so grainy. It's my camera's not the best. Also, there's so many bacteria in this sample that it kind of gives the footage a grainy feel to it. The large comma-shaped things are a ciliate that I also recognize from the literature. I can't remember what it's called. If I remember, I'll put it in the description. And that ciliate is an organism that's convergently evolved alongside Thiovolum majus, and it creates veils too, but instead of dragging sulfide upwards and oxygen downwards, they use their veil to take sulfur oxidizing bacteria deep in the sediment and to transport them upwards for them to feed upon. So, these being eukaryotes, they predominantly live in oxygen-rich habitats. They need oxygen to metabolize. And their food exists in an entirely different environment that they cannot survive in. So by creating this veil, they're able to live in their ideal environment and then drag their food from an inhospitable environment upwards towards them. So it literally is like a tractor beam. But they're not the only organisms here, though. I think that they're engaging in some sort of like mutual exploitation, where the Thiovolum majus and the ciliates and all these other organisms are coming together because this current, this cyclone, helps to concentrate resources that are more diffuse in the surrounding water. So things like iron or molybdenum or manganese, things that might be difficult to forage for or concentrate along this band by the constant shifting of the water. Like, why move and search for resources when resources are delivered to you? So I'm going to zoom in a little bit further. Hopefully we could see a little bit more of this community's composition. So this is really interesting. Now, the thing though is that when you take a community like this from sediment that you've collected or a water sample, you're disturbing it. You're changing the spatial orientation of these organisms and how they configure themselves in their natural environment. So maybe this isn't their natural arrangement. Maybe this is essentially what happens when you jostle them all together. But it seems like you have filamentous organisms, likely cyanobacteria, or maybe some sort of photoheterotroph, like an anoxygenic photosynthesizer, maybe chloroflexi. I've heard that they tend to live in sulfur-rich environments, and they live alongside sulfide oxidizers. I could be mistaken, though. But they seem to be forming a sort of matrix which is that clear gelatin-like material. So the chloroflexi and all these other organisms that you could see as transparent rods and spheres and dots all across the screen, they're all coming together to create a biofilm, which forms a platform for these communities of organisms to attach to. I'm going to zoom back out, just take a look at the overall structure one last time. And I just want to show you guys how expansive this is. And again, you have predators coming to it as well. That little, some of those like little jumping things. Like, I'll try to catch it. Like that large sort of jumping object that's kind of spazzing about. That's Halteria. That's a ciliate, and it's likely feeding on these guys. But let's just go up and down this structure and just view it. Sorry for the blurriness again. But look at that. That is so incredible. In this area, it looks like it becomes a little bit more diffuse. Oh, look, you can see that these guys are navigating. They were probably like loose in the sample when I jostled them around. And now they found their ideal environment where sulfide and oxygen come into contact, and now they're building a structure on that barrier, or on that border between those two habitats. So this is being made in real time, potentially, if I'm interpreting this properly. Let's see it really quick. On a final note, before closing out this video, I also want to bring up this Luxodes, which is a ciliate. It's foraging right next to the veil. So this is a great example of other predators being attracted to these structures. This whole entire thing operates kind of like a coral reef. So they are essentially building a new home, a new habitat for other organisms to live in. That's a great shot. You can actually, like, see him spinning away. See, there's a great example of the current. I'm sure as I comb through the footage, maybe I'll see a couple dividing, too. There we go. You can actually see the bacteria that form up the veil. All those little particles. Some ciliates. Not sure what these guys are. Maybe Halteria. Little cyanobacterium. The long bluish green filamentous thing. But yeah, that's enough for now. I'll talk to you guys later. Take care. Oh, and a diatom. Okay, take care.